It's a great pleasure and honor to be here. We had an absolutely exciting scientific symposium yesterday, and I'm going to deliver my message from north of the wall, but certainly many of the people who will be on the panel and who we heard from yesterday could give the equivalent talk if they came to Canada. So I think you're in for being informed on how genetics is really revolutionizing the understanding of ourselves as humans, but also how it's being applied in the clinical setting to understand and provide better answers to families. So my opening slide is really to address the question is, why is genetics important? And the answer is that DNA is a blueprint for your life. Each of us is comprised of roughly an average-sized human 60 trillion cells. And in every single cell in your body, you have the same complement of DNA. And all of that DNA is what we call your genome. So I use the term genome throughout my presentation, and you'll hear it from others too. That's a complete set of genetic instructions in all of the cells in your body. And you inherit half of that DNA from your mother and half of that DNA from your father. Like it or not, you are one half of each of your parents. And as children, we grow up fighting that, and particularly when we go through adolescence. And then after you get married, you can ask your husband or your wife, and they'll say, he's just like his mother or just like his father. And the reason for that is we're pretty darn hardwired. The DNA is your blueprint, but blueprint is the key word because the DNA stays static, but the genes that come on and off from the DNA can be altered. And your environment, the things you eat, the decisions you make, the temperature that you're living in, be it in the north or the south, influences how your genes come on and off. So most genetics research is trying to identify the genes involved in specific diseases. There's over 6,000 different diseases that bear different names and have different clinical outcomes. And as I'll show you a little bit later in my talk, the advances in human genetics, mainly from the technological and information science perspective, have allowed us to find the underlying genetic factors or disease genes in roughly about half of those genetic disorders now. Okay, and this has all come about largely in the last 15 years or so. And I'm going to tell you a little bit about the advances in genetics in autism, and we'll talk about more how to use this information. So on the opening slide, it really shows a fertilized egg. This is an egg and a sperm and a series of sperms. One sperm will win out in the battle for fertilization. So you start your life as a fertilized egg, and then it keeps doubling and doubling and doubling. And this is really the essence of life until you pass away and your genome goes with you. But as I said earlier, you pass that DNA on to your children, and it's really the legacy of the humankind. It's a set of genetic variations that are passed throughout the human species that actually are our common inheritance. So I love this picture on the right-hand side. It captures all the different images of representation across the globe. And if you were to compare the DNA sequence between any two people on that slide, be they black or white or yellow or mixed in between, the genomes are 99.9% identical. So you would find roughly a one-letter genetic change every 1,000 or so nucleotides along the genetic backbone of the chromosomes. And what that means is we all share a very common humanity. Most of the variation collected in our genomes occurred when we were a common species in Africa. Perhaps we didn't start migrating out of Africa as a species maybe 100,000 or 70,000 years ago or so. That's in debate, but roughly 100,000 years ago. And I'll talk about this in my presentation, but as an individual, you actually carry only roughly about 100 different genetic changes compared to those that your parents carry. Okay, so the replication of DNA is very high fidelity. And when you have a new genetic change in a specific gene, it can lead to a specific clinical outcome. And I'll show you in my presentation now, the community of scientists have identified specific genes that are involved in brain development that are also involved in autism. And that's been the big advance in our field in the last, I would say, 15 years. We really didn't know about any of the genetics of autism, certainly in a specific manner, up until the advances of DNA sequencing technology that I'll tell you a little bit about. So I'll tell you kind of the status and my interpretation of what's happened in the field of autism. And these are the 2 take-home points that you should draw from my presentation today. The first is that there's enormous variation in clinical presentation in autism, and we often call it the autisms or autism spectrum disorders. You'll hear over and over again, probably tonight, that every single individual that has autism is different. They're like snowflakes. So certainly we now know that autism or the autisms are collections of rare disorders or conditions that share features involving brain cells. And the brain cells are called neurons. I'll use the term neuron or brain cell throughout my presentation. And secondly, there's enormous variation in the genetic factors involved in autism. Autism is a common clinical presentation, but there's lots of ways to become autistic. And genes and changes in genes, alterations in genes, are really the major underlying contributor to autism. So we know now that there's certainly dozens to perhaps hundreds of genes that can be involved. Some of these are high impact genes. If you carry a genetic alteration in a single gene, it's sufficient to cause autism or ASD. And as a community of scientists and clinicians, we think that understanding these genes and the factors that influence them, in some cases your environment, may lead to precision treatments to better help families and individuals who are confronted with autism and need to have a medical management plan. So throughout my presentation, I'll talk a little bit about trees and branches on trees and leaves on trees. And this is a very famous painting from the north, from Canada, by Canada's most famous artist, Tom Thompson. And it shows what Canada looks like in its autumn season in October. And I like to show this picture when I talk to crowds that are interested in autism, because the whole idea behind identifying the genetic factors in autism, humans can change, just like nature can change its colors. And if we can identify the genetic factors involved, maybe we can change the environment that influences those using interventions, medications, and change the leaves of the colors to adapt better to that individual's needs. That's really what we're aiming to do here. So why do we perform research in ASD or autism? And these are the most important points. The truth will be found in science. We've known now for 50 years, we hear different things come up in the public media, discussions about what causes autism and what doesn't cause autism. We've heard things like refrigerator parents not raising their children, giving them love and affection, to vaccination hypotheses. And every time we need to test the hypothesis and kind of question these things, what's real, what's false, you have to turn back to science and do controlled experiments. And I'm going to give you a lot of results in my presentation, but these are the basis of millions of dollars invested in research to try to answer the questions that families ask us to answer. And we've done surveys in our Canadian families, and the questions the families with autism want us to solve first, and most importantly, using science, are what is the cause of autism? Why did autism come about in my child, for example? Secondly, what can we do about it? What are the next steps? And third, and this has come up over and over again, they want to know how else they can help other families. And this is why they enroll in our research studies. Second, we think and we have evidence now that early detection of autism and related conditions may benefit the health outcomes. So a genetic diagnosis can actually precede the clinical presentations of some of the symptoms, and I'll talk about this later. Better understanding of the causes of autism will benefit the medical management. Autism really was an enigma. I'll use that term in my next few slides, a black box. We really didn't know much about it even better part of a decade or so ago, and things are changing with the genetic advances. And you'll hear a lot about this concept of precision medicine, individualized or personalized medicine. This is individualized approaches may be the way forward in autism and many other medical conditions. By understanding your genetic blueprint, it may guide and tailor the drugs that you need to take in the right doses to actually treat different attributes or traits or disorders or diseases. In many cases, of course, this is going to be guided by the genes that you have in your genome. And then last, and I think also very, very importantly, is studying autism by studying individuals with autism and the whole autism presentation, we have a much better understanding of human development. And the reason for that is the core features that encompass an autism diagnosis involve cognition, speech, social interaction, and behavior. And in fact, these are the attributes that make us most human as a species. These are the things that helped us crawl out of up the evolutionary ladder, the ability to socially interact. So in fact, by studying people with autism, we're studying ourselves as human beings, understanding ourselves, where we need to go as a species, as families, as individuals. So this is one of the very first genetic diagnoses we used using a whole genome sequencing approach. This was the cover of the Toronto Star in 2008. And this young lad came through the clinics at our children's hospital, the Hospital for Sick Children, and he went through 8 different clinics. Lots of money spent on testing, lots of blood draws. The family was very upset. And he did have a diagnosis of autism, but he had a lot of other complications. But they didn't know, the family didn't know why. And it was really through using the genetic testing that I'll show you a little bit later that we, in fact, found for the first time a region on his chromosome 16 that was deleted. This is what we call a copy number variation. In fact, it's a segment of DNA that's 680,000 letters long that deletes 17 genes, and 70% of those genes make proteins that are involved in how the brain functions. But as the title points out, solving the puzzle of this son's autism soothed the family because it gave them an explanation, an end to their diagnostic odyssey. This is the cause. And in fact, this is the most frequent change that we see involved in autism in our Canadian cohort, and it's the same also in Australia here. It's typically a spontaneous change that occurs, and it accounts for about 0.7% of autistic diagnoses. So I took 4 quotes to help explain what autism is that really struck me when I read these. And these are, the first 2 are extracted from a book, Dr. Peter Zatmari, who is also a Canadian. He's a child psychiatrist. I would say he's the father or grandfather of genetics research in Canada, but he would be mad at me because he's not that old, actually. But this is his book, and he's seen thousands of patients. And his first quote is, "It all depends on the way you see things," said the woman on the other side of the little table in my office. "What can seem like a disability one day can be a talent or a gift on another day." And secondly, what is so tragic is that the impairments in social interaction, in communication, and in play strike at the very heart of what it means to be a child. And so families want to know why this happens. And as I'll show you, genetics is a major factor. And then these are 2 quotes from non-Canadians, but very famous scientists and physicians in the field. And the first is from Uta Frith, and she says, "People with autism cannot see the forest for the tree, but they see the trees in exquisite detail." And second is Michael Rutter, who introduced the concept of genetics along with others in the field of autism studies. And his quote is, "Examination of monozygotic twins concordant for autism show enormous clinical heterogeneity even when pairs share exactly the same segregating alleles or genes." And I'll explain what this means in a moment. And then what I didn't put in writing is a quote that I heard from one of my friends who has a severely autistic boy, and he told me probably about a decade ago, for him, having his son having an autism diagnosis or having autism meant he was trapped in his terrible twos 24/7, 7 days a week. And they didn't understand why he wouldn't communicate or interact with the family, and they wanted to know, what they could do about this. So the stories of autism are as diverse as the snowflakes that fall in the north in Canada. That's my point. Now, what many people don't know is that there's many coexisting mental and health conditions that come along with autism spectrum disorder. And this is a study that was published in the province of Ontario, which is the province that Toronto resides in. And it was, I think, published in 2013. So we see attention deficit hyperactivity disorder in the Ontario population in about 14% of autistic individuals. In fact, in many other studies, it's upwards of 50%. Sleep disorders, 15%, learning disability, 22%, depression, 28%, and anxiety, well here it's anxiety actually, 45%. And then if you look at the health conditions, things like digestive system disorders in near to 50% of individuals, and brain and spinal challenges, and in particular epilepsy or seizure presentations in 50% of individuals. So there's a lot going on here. There's a lot of complexity in the clinical presentation. And if I forget to mention this a little bit later, many of us in the field think that perhaps treating these so-called comorbidities or complications that come along with autism are the best entry point into new treatments, because drugs may exist for these. For the actual core features that impact, that lead to autism or that are involved in autism, there's no medicines, no pharmaceuticals that are currently designed to target those core features. And I'll come back to this at the end of my presentation. So back in, I think it was in the 1970s, the first genetic studies were introduced, and I call this really to the genetic or genomic enlightenment in ASD. Genes have been the key clues to crack the autism enigma. And the first came by the study of monozygotic twins. So these are twins that have the same genomes, identical genomes. They may have different changes that occur somatically in the first few cells of division, but actually the genomes are identical when they start. In studies that were done at the population level, the first studies showed that 90% of monozygotic twins, if one child had a diagnosis of autism, 90% of the time the second child would also have autism. And subsequently, there's been other studies using different criteria that showed up to 50, 60, 70%. It was always in the 50 to 90% range. And then if you looked at dizygotic twins, which share half their genomes, that rate drops off a lot. This is a classic signature that genes are involved in any genetic disorder where you do this type of twin comparison study. So the way you take this is, it doesn't mean that 90% of autism is genetic. It means that in this study, this is the rate of concordance in the study, but the take-home message here is that genes are involved or DNA is involved. And then secondly, in roughly 18% of families, if the first child receives a diagnosis in 18% of cases, and again, this ranges in 10 to 20% depending on the study, a second child will also develop ASD, compared to the typical roughly 1% in the general population. Okay, so again, that means that genes are involved. And to cut a long story short, over 15 years of research, many laboratories around the world, and we made some seminal contributions early on, identified these copy number variations or CNVs, like the chromosome 16 change I showed in the boy in the Toronto Star article. And now more recently, using high-resolution DNA sequencing technologies, changes in single genes that can cause ASD. Now again, these are all rare, and they occur in less than 1% of the frequency in the population of autistic individuals. Okay? So as a community of scientists and medical researchers now, roughly 100 or so ASD-relevant genes have been identified. And all of these encode proteins or molecules that are involved in brain function. So these are just a couple of images. These are monozygotic twins here. And then this is what a copy number variant looks like when you image it under a microscope. You see a segment of the DNA actually missing. And then if you were to look under this kind of graphical image, you would see a change or a genetic alteration, a mutation, if you will. So a change in a DNA sequence that leads to a clinical outcome, geneticists call that a mutation. So you hear that term throughout the presentations tonight. So all of this came about really because of incredible advances in the genetics and genomics technologies. The state-of-the-art technology in the 1960s, and in fact still is the predominant test that's used in most clinical diagnostic laboratories, is to do what's called cytogenetics or chromosome karyotyping. You stain the chromosomes and you look at the morphology and you count for numbers, or you look for structural changes based on changes in length of the chromosomes. And at our hospital, this still is the #1 test that's done, in particular for cancers. This is a male because there's an X and a Y chromosome here, and I think this is a typical karyotype, but if you were to see trisomy 21, for example, that would often lead to Down syndrome. So karyotyping now in the research setting costs about $300 a scan, and this will become relevant in a second. And it detects large structural changes in chromosomes, as I mentioned. And in fact, these large changes account for roughly 3% of the cases that we've studied in our Canadian cohort. And then there was a lot of years of molecular biology, a lot of Nobel Prizes awarded for DNA sequencing and things like PCR. And then in the 2000s, these what are called chromosome microarrays were developed. This is a high-resolution image the size of essentially a quarter. It's actually a microscope slide cover, essentially. And dotted on this are stretches of DNA. There's actually 2.7 million stretches of DNA that are targets across the human genome reference sequence. Now you can take DNA from a patient with autism or anyone from the population and look for these copy number changes by doing a hybridization experiment. So we helped develop some of these arrays in the early days. This is now, the chromosome microarray test is the standard of care test in North America, and I think also here. We'll hear about it. Yes, it is in here, in Australia, and in many places in the world. So this will be the first tier test for autism and neurodevelopmental disorders. It also costs roughly $350 a scan. This is in the research setting. Some of the people are going to say it's more in the clinical, but I run a research laboratory. And I point the prices out because this typically what happens is the newer technology doesn't get implemented in a hospital laboratory. Even though it's a higher resolution test, we can scan the genome and find lots of additional changes compared to karyotyping until it comes down to the same price, because you have to build a health economic argument to get the government to pay for it. But here it detects 7 to 10% of ASD cases. It explains them, these copy number variants. And then this is actually from Illumina, the company, which is the world's leading DNA sequencing technology company. One of their earlier next generation sequencing machines that generates these clusters of sequences where you're sequencing by synthesis. And in fact, now there's a technology called Exome you'll hear a little bit about. And what that does is you use a microarray to pull down all of the little segments of DNA that encode genes. So 1.6% of the whole genome sequence encodes the protein coding regions in genes. So you pull that down and then you do the sequencing. That's roughly, well, I think last time I made it $750 an exome. Maybe it's $500 or so now. But we think that maybe in 2020, this will become the standard of care, first-tier test along with microarrays, as long as the pricing comes down into in the same range. And that will probably happen in Australia sometime relatively soon. And Canada is at the same pace as Australia. And in the States, there's different payers. It happens at different times. But I'll show you that this is a much more sensitive test because it looks for the very small level sequence changes in the genome. And we now know that this technology can capture about 7 to 10% of the explanations of ASD. These two technologies, microarrays and exomes, are largely exclusive. So they're finding different genetic alterations. Now, the state-of-the-art research technology is called whole genome sequencing. And this is what we're using in our research laboratory, and we use the Illumina technology also. And it, for roughly $1,000 US, can sequence all of the DNA in essentially a 2-day experiment, and you can find essentially all of the genetic variants in a single experiment. No technology will ever be perfect, but this is pretty darn good. And we decided in my laboratory and in my team that it was good enough that we would embark on a sequencing project to look at thousands of families with autism. And I won't go through the details. We talked a lot about this technically yesterday in the symposium, about what these different repeats are and these different types of genetic changes and how they're involved in autism. But ideally, what's happened over the years is laboratories use these tiered testing approaches using different technologies. Each one happens in kind of an incremental order. But now, I think the whole field of diagnostics will be turned on its head because, again, in a single experiment, you can find all of this genetic variation, essentially all of it. So you really get 3.2 million different genetic changes you can identify in a single experiment. It's a lot of data. So as scientists, we're actually struggling with how to interpret all of that data. But in fact, it's not that much data. We were involved in sequencing Craig Venter's genome in collaboration with his company called Genomics. And I remember at the time when Craig Venter had his genome done, we handed his genome sequence to him on a CD-ROM. And in fact, you can carry the genome sequence on a USB key now. It's quite likely that you'll carry it around on your phone when in the next few years or so. In fact, Illumina has a project to do this. So it's quite incredible. We've come from sequencing the genome as a $3 billion project. Venter's project himself cost $70 million. When we started our autism sequencing project, we were doing genomes at $7,000 a genome, and now we're at roughly $1,000 a genome. So with Autism Speaks, which is an international autism agency, we embarked on sequencing lots of families to try to find all of the genetic factors involved in ASD. And I won't go through all the details, but we started this wholeheartedly in about 2013. And it was a partnership with Autism Speaks, Google, and the Hospital for Sick Children. Our goal was to turn into digital information every DNA sample that had been collected by Autism Speaks over their 20-year existence. So that scientists didn't have to sequence themselves. They could go to the cloud, pull down the sequences, and do their experiments. As simple as that, to turn DNA into digital information. So a lot of the samples were in fact Canadian because we had invested significantly in genetic research. And we're now following over 4,000 families across, this is Canada here if you don't recognize it. And these have all been enrolled through our Research Ethics Board consented protocol. And the major questions the families and our ethics panels at our hospitals wanted us to address were twofold. One is, where would the data be stored? And how secure would it be? And when we started this, no one really knew what the cloud was. I remember we had to call in Google to explain to our hospital administrators what the cloud was. And they didn't believe us. They had to hear it from Google. It was more secure than storing it on your laptop in the laboratory. And secondly, what would you do if you found secondary or incidental findings? So while you're looking for genes involved in autism, what if you find a gene involved in breast cancer or something else? How are you going to deal with that information? And what are the infrastructure in place to help the families? Anyways, we dealt with all these. The families altruistically enrolled in this study to address the questions that I mentioned earlier. Why? What can we do now, and how can we help other families? And they agreed to put all of their information on a cloud, and I'll come back to this a little. Well, actually, I won't. Anyone can go and get this. Yeah, I will. Anyone can go back to this data, but you have to have approval from a data access committee. Okay. And if your question has to do with autism, you can go and get the data, but you have to be approved by a committee and sign documentation. So the data is indeed protected to the research community, and that's very important. So anyways, we've now sequenced 11,500 DNA samples, and the site of the sample based on the postal code of their enrollment ranges from the west coast of British Columbia to the east coast of Newfoundland. Probably 60% of the samples are from Ontario. This is Toronto down here. There's also provincial projects involved. The province of British Columbia has a targeted project around diagnostics and translation of the information. Ontario has the what's called the Province of Ontario Neurodevelopmental Networks. We talked a lot about that today in meetings, modeling projects here in Australia. And then Quebec also has a provincial project. Now, I should say many of the samples also came from the United States and elsewhere in the world, but it was mainly a North American project. We spent a lot of time getting the data into a form that users could actually access it, and I won't go into any detail here, but if you were an autism researcher and you wanted to download all of the information, you could do that, and a few people have. When we transmitted the first tranche of data to Google, it was the largest data set they had ever received in their history. This is the complexity of the data we're dealing with. We have all the clinical information in the cloud. You can search by genes, and the idea is to make the information available to doctors and the families actually participating so that you can match your genetic findings to someone else and then share stories if your child has autism and a genetic change, and find if another person somewhere else in Canada or elsewhere in the world, perhaps in Australia, and share your experiences, your life experience, what works for kids that have the same genetic change, or adults may work in others that have the same genetic changes. So right now there's 200 researchers from 59 institutions from 17 countries. using the data. And there's a project called the Global Alliance for Health. In fact, Catherine North here in Melbourne is the current leader of the Global Alliance. And it deals with information across all different genetic diseases and disorders. But we've actually made some of the more interesting genetic information available to the larger community of researchers dealing with, for example, epilepsy or anxiety, who want to also access data. The idea is to get the data out to somebody who might have a really brilliant idea, the student in China or in India or Canada or in Australia, who can't afford to do the experiments themselves, but they have a really good idea. And they want to take it to the cloud, and they can do it for free on someone else's dime. And that's happened multiple times now. So I've summarized all of the genetic observations in a decade from scientists around the world in this one slide. This is probably a little bit more for the techies or the geneticists, but I think it's important to set up the remainder of my talk. So we've now identified what are called de novo or spontaneous or rare inherited changes in single genes that can be sufficient to cause autism. Okay. In some cases, there could be other common genetic variants that occur that modify the impact of that initial high-impact genetic change. These are called common variant modifiers. And as we look at large populations of individuals, we'll be able to better map the influence of these. It is very important, but these are really act in kind of a role of modifying the extent of the autism presentation. Most of the known ASD-relevant genes are involved in other neurodevelopmental disorders. Things like epilepsy I talked about, intellectual disability, and other things. Many of them are actually on the X chromosome. I think I mentioned males have an X and a Y chromosome, and females have 2 X chromosomes. So females can be missing one of their genes on the X chromosome and still have a backup. But if the change happens in a male, they don't have that backup, so they're missing entirely a gene. So we think that this is one of the reasons we see this preponderance of this so-called sex effect of 4-to-1 male to female bias in autism, explained in part by the genes that have been identified on the X chromosome. Most of the genetic alterations are these copy number variant deletions that go from 2 copies of a gene in a genome, or a so-called loss of function mutation or genetic change, so that only 1 copy of the gene/protein is functional. Okay, so it changes the equilibrium, the dynamics, the homeostasis in the cells. So that's one of the changes, and I'll come back to this a little bit later. Of the known genetic changes, they do not seem to be environmentally induced, but in fact they occur randomly. So we've looked across populations from samples collected around the world, and the rates of new mutations or changes that accumulate in autism versus any other population, including controls, are the same. It's just that the genes impacted in individuals for autism are these ones that are involved in how the neurons function. Whereas each of us carry the same number of changes. I carry my 100 mutations that are different from my parents, and they contribute to what makes me unique. None of them hit the autism genes, though, so that's why I don't have autism. Okay, so far all of the genes, proteins are somewhat involved in the neuronal or brain cell synapse function, but acting in many different diverse ways. So come back to the tree analogy. This is actually a photograph I took. It's a little bit out of focus here, but I liked it because when you look up the trunk of the trees or the neurons, you can see how the branches and the twigs and the leaves kind of interact. This is exactly what happens in your brain. Okay, neurons are like trees in your brain, but instead of just having 2, you've got billions of these things packed into your brain that have to work and function and develop and communicate with each other. So seeing the differences in the trees and branches is essentially what we're doing when we can actually go into the brain and look using imaging, for example, but more recently, I'll show you an image on my next slide, we can actually take cells, we can take blood cells or skin cells from an individual, from anyone in the population, and then use a differentiation approach. These are called embryonic stem cell differentiation, and turn them into brain cells. So one of the challenges, or heart cells or kidney cells, one of the challenges in autism research is you can't directly access any individual's brain to do a study, but you'd like to model in a petri dish or in a plate the genetic changes in that individual. So we can now take their skin cells and do what's called induced pluripotent stem cell differentiation, and then turn them into brain cells and model these things in actually the genetic change in the petri plate in the laboratory. This is unbelievable. We couldn't do this 10 years ago. So genes such as CHD8, Nerexin1, SHANK1, SHANK3, ARID1B, SCN2A, these are the top autism single genes that have been identified so far. And I just have an example what SCN2A does. It makes a protein that spans the membrane, and it's involved in shuttling molecules back and forth, sodium molecules back and forth across the cell membrane. And if you have specific genetic changes in this gene at one site in the gene that makes part of the protein, it leads to a form of epilepsy. And then towards the other end, it leads to, the changes lead to a full-blown autism clinical presentation. It's really amazing to me when we think that there's any specificity at all when you have a genetic change that affects a protein that's present in 100 billion cells that are crammed into your brain. But in fact, there is. And I think this is the reason we see this overlap with many different clinical conditions, because there's lots of different ways these neurons can actually communicate. In this case, the SCN2A change leads to a reduction, so from 100% of protein to 50%. Okay? But then the complexity is so significant. In fact, there's trillions of different, I think it's 90 trillion different synaptic connections occur in your brain. And the brain is the organ that, it's the most important organ. I argue with the cardiologist at our hospital that it's more important than the heart. But I think there's, the brain is the organ. It's been fine-tuned through evolution to be humming. It has to hum. This is the organ that has developed AI approaches and computer programming and processing units that try to outcompete the brain. It will never outcompete the brain because the brain conceives these other ideas. But in fact, it's these subtle changes that lead to different rewiring of how these neurons align and communicate information. And that's the difference that we're seeing in the forms of autism that we've studied so far. And this is my picture is worth 1,000 words. This is the first image we had where we looked at these differentiated stem cells that turned into neurons. This is back in 2012, I think it was, where we did this type of experiment in lines that came from Nerexin1, genetic alterations, SHANK2, SHANK3. And typically what you see at this high imaging resolution is a change in the volume or density of the twigs and leaves and branches on the trees. In this case, it's the neurons. So you can zoom in and actually measure the density of the different dendritic spines, and they're shown here along the neuron, and calculate that. In fact, there's just a reduction, roughly in about 50% in this case, that we see in an individual that has ASD versus a control. And the controls are typically parents, gender-matched parents that we do the same experiment in. Okay, in some cases you see an overbranching, a higher density of these of the branches. In some cases it's lower. It depends on the genetic change. But that's the variation happening because of the genetic alteration at the physiological level. And in fact, this picture came from an individual in our genetic study where we found one of these genetic changes. And the diagnosis impacted her, but it was really this picture that set up the science in the next set of experiments. So based on what we know now, these are the genetic categories of ASD. We talked yesterday a lot about different definitions and terms used. These are the ones we're using in Canada. So-called clinically defined ASD occurs from roughly 4 to 5% of cases. These are the chromosome changes, including Down syndrome. You can often see ASD. Changes in neurofibromatosis, tuberous sclerosis, PTEN-associated mutations. These involving children who have macrocephaly, larger head circumference. Some of these microdeletion syndromes, 22q deletions. And then also where there can be an environmental influence, including valproic acid. And then in roughly 20% of cases, these are what we call molecularly defined. So these are changes detected using the microarrays and the sequencing approaches, including in some cases chromosome changes and these ASD relevant genes that I showed on my previous slide, including SCN2A. And then the copy number variation, deletions, and also duplications. So I talk a lot about deletions, but in fact there's a region, for example, on chromosome 1, Q21. If you have a deletion, you don't have autism. You have a type of cardiac problem. But if you have a duplication, so 3 copies, you can have other complications, but you have autism. So it depends on the dosage or the number of genes, depending on the function of the gene. And very importantly, still in roughly 75% of cases, we don't know what the genetic cause is. Okay, it could be, in fact, that there's multiple genes in combination, and when we have a larger sample set to compare against, we'll be able to statistically kind of tease this out. It could be there's genetic changes we haven't found yet, because the whole genome sequencing is good, but it doesn't get everything. It could be common genetic variants, or it could be that there are environmental changes that are impacting perhaps the same genetic pathways that I told you about in my last couple of slides. But we've come to get this 25% so-called clinical yield in the last decade, so we've come a long, long way, and we know a lot more now. And this is making its way into the clinics, as I'll show you in the next few slides. So I selected 5 examples, really out of hundreds, where we've found these genetic alterations in our research studies, and then the data is communicated back to the families through a genetic counselor or through the clinical genetics clinic at the Hospital for Sick Children or elsewhere across the country. As I said, these are 5 examples. So the first one I selected was based on a genetic change that was spontaneous, so was not found in either of the parents. It leads to what we call as a frame shift mutation, so it leads to a truncation of the gene, so the protein actually kind of falls apart after it's made in the cell. And the impact is, in fact, this same gene had been found previously to be involved in another genetic disorder called Wiedemann-Steiner syndrome. So we know there's 100 different genetic disorders that carry different names that also exhibit autistic traits. And in fact, probably 40% of Wiedemann-Steiner syndrome individuals have a form of autism. So when we made this observation, this explains the autism, but she actually also has Wiedemann-Steiner syndrome, the same symptoms you see in other individuals. And going back into the cohort that we had studied across Canada, we found certainly, I think it was a half dozen other individuals that were unrelated that had genetic changes in the same gene. Okay, so this is really subcategorizing autism in a very specific form, in this case called the WSS form, with genetic changes in the KMT2A gene. This is the second family. This family was collected in the province of Ontario about 30 years ago. It was one of the first families that Peter Zatmari had collected many, many years ago as part of the genetic studies. And we studied it using different technologies and ultimately found what we think is the high impact genetic change using whole genome sequencing. In the RAB39B gene, this leads to a termination of the gene product and protein. Now in this case it's maternally inherited, but it's on the X chromosome. Okay, so mom's got the backup X. So she actually has a milder form of autism. It's often called atypical autism or broader autism phenotype. But when that change is passed on to males, they have full-blown characteristics shown here in the black boxes. And again, passed to her daughter, a female carrier, as a subclinical presentation. So this was interesting because this gene had been implicated in a form of movement disorder called Parkinson's movement tremors. And getting the diagnosis was very important for the medications that these individuals were taking. And it also explained 30 years after they enrolled in the research study why we found there in a CSI way that the explanation of the autism in the family. So it was really critical for the family to know this. So we had to contact them and go back and share this information from a family we studied some 20 or 30 years ago. The third family was a 1.17 megabase, so that's 1,170,000 nucleotide or chemical letter deletion in a region on the X chromosome. This is a de novo or spontaneous mutation. It's an X-linked region. Now, I talked about this at our symposium yesterday. We found lots of these changes, and if you carry a deletion in this region and you're a male, everyone we've seen so far has autism, so it's quite predictive of an autism outcome if you're a male. But 99% of the families we've studied with this genetic change, the change is passed from an unaffected carrier female to an affected male. This was the first instance where it was a de novo change, and that's really important for estimating the frequency of likelihood it would happen again in the same family. It returns back then to the population average of, in Canada, it's 1 in 65 or so, 1 in 66. Okay, so that's very important genetic counseling information for the families to have. And from the biological perspective, this is the first time we found a molecule that doesn't encode a protein. It's actually an RNA molecule that's involved in regulating other genes' expression. So it gave a real new clue into the etiology of autism and perhaps new targets that we can target via new medicines. And the fourth family has two ASD individuals. We found two different genetic changes. One is inherited from the mom. This is a gene that encodes a protein involved in a form of epilepsy. And then in the firstborn boy, we found a spontaneous or de novo change in the thyroid hormone receptor alpha gene. And around the same time we had published, well, this is actually published first in 2015, sorry. There were other individuals with autism and also intellectual disability and a mouse model, actually a mouse that had the exact same mutation as this boy here, and they exhibited features of reminiscent of what autism would look like in mice. The mice didn't like to socialize with each other and interact. But in fact, in most pediatric hospitals, thyroxine, which is the treatment here, is used hundreds of times every day. And the individual was given thyroxine treatment, and it's actually led to a significant impact on his energy and metabolism and a very positive outcome in his management. Now, the point here is that we needed to know these genetic changes are different between the siblings, so you would treat them in different ways. Okay, so this again is an example of the precision medicine approach. Knowing the genetic change will impact and perhaps influence the treatment, the medical treatment of that individual. And then the last example is also on the X chromosome, and this was maternally inherited, and we found this very high impact. Again, if you have this genetic change and you have a family history of autism, in almost every case there will be an autism outcome or something that looks like autism in a second-born child. But in this case, we could identify it very early on, in fact, pre-symptomatically, and enroll this individual in physical or behavioral therapy. And it had, we think, a sufficient impact that his clinical presentation is much less so than his older brother. So this is really the goal of using genetics for early identification, so you can identify someone accurately and enroll them in early intervention protocols, and perhaps ultimately give them medicines once the medicines are developed. I just have a few more slides here. I went back into the archives and dug this one out from 2010 because it's actually a nicer figure than our latest paper that came out just last year. And I showed it because we predicted that probably all of these genes would be involved in autism, and most of them are in some way. And what you're doing here is you're mapping a particular gene's putative cellular function in relationship to any other gene/protein in the cell based on the number of times they interact in the same genetic network. Okay, so this is called genetic interaction pathway mapping. And so if two names of genes are close together, it means they work closer in the cell to each other. So for example, if we find, I'll just pick an example here of SYNGAP1 or GRIN2B, and there's a genetic change that knocks down 50% of that in the cell, you can take different therapeutic strategies. Try to increase that gene/protein back up to 100%, or maybe you don't have to get it, maybe up to 75%, or target the genes that are just downstream but interacting with it, if there's a known drug, for example, that works in that system, to increase. But you need to know what the genetic change is if you have to increase or decrease. And then you use this kind of approach to target medicines based on the molecules that are present or altered in the cell. So this is exactly what drives any of the new drug development that's happening in the field of autism, and perhaps probably in most other disorders now. Again, a precision medicine approach. And in fact, in the last 15 years, many new medicines have been developed that are being tested either in mouse models. So I mentioned that you can model mutations that are found in humans and mice, and we talked a lot about how to actually test mice for autistic symptoms yesterday. You can do these induced pluripotent stem cell neuronal lines and check how new medicines alter the proteins and genes involved. Can you increase the dendritic branching, the leaves and trees the leaves and branches on the tree? And these are the list of the drugs that are going through different stages of human clinical trials based on the genetic discoveries. And I won't go through all of these, but they're targeting different genetic pathways and different genes that I've talked about throughout my presentation. So I think there's a lot of hope for the community that the genetic advances are, of course, impacting diagnosis, but also informing on development of new medicines that I have a lot of hope for. I really do believe that sometime in the future there will be new medicines that are going to be available for doctors and families and individuals that have autism. My second last slide. I thought we'd show this. In fact, it was shown yesterday, and we'll talk about it, I think, in the discussion. This is the recommended genetic testing protocol in Canada now, and it's very similar here in Australia, as we heard yesterday. So if there's a diagnosis or a family history of autism, there needs to be a full family history taken. And I forgot to mention this at the start, but the first question a doctor asks you is, do you have a family history? What they're asking you is, what are your genetics like, right? That's really what it is. So can you imagine in the future, you're going to give them the answer by handing them your genome sequence and also describing your experiences. So also physical examination. And so I'll start on the right-hand side. So you have a diagnosis of autism plus a more complex set of complications that are involved, so-called complex ASD. Then you would use targeted genetic testing, perhaps karyotyping, perhaps looking at specific genes. But ultimately, right now, the first-tier test is the microarrays I talked about. And probably pretty soon, it'll be whole exome sequencing. And ultimately, it'll be whole genome sequencing once the prices kind of come in check and the testing has been done to set these up in the laboratory. If you have the so-called essential ASD, which is largely overlapping with the 75% of uncharacterized cases, we're recommending still the protocol of testing of microarray, for example, Fragile X testing. Girls with intellectual disability, we look at this specific gene called MECP2. And then if there's other kind of morphological differences in, say, head size or other things, you can check specific genes that I talked about earlier. But this is the protocol that exists now. Okay, and I think we'll talk about this in the discussion. Ideally, we'd like to go and do whole genome sequencing like we've done in this large study across Canada. But that will come in the future as the prices come down, as the understanding of the utility of the technology comes into full practice. So just in conclusion, these are some of the big questions that we discussed yesterday and that I think about quite often in our research study design. When should genetic or genomic data be used? Different people will have different opinions. In Canada, if the individual comes to medical attention and genetics might help, we try our very best to offer genetic testing. And I showed in the last slide how we do this clinically. And if we can't do it clinically or we don't get an answer, we enroll them in our research study. And that's really driven our research study to sequence the genomes of thousands of Canadian participants who want to participate actively in these studies. Secondly, are there truly core pathways underlying the core ASD features? And what will be the best therapeutic approach? Will it be physical or behavioral intervention? Will it be a drug, a small molecule? It could be DNA itself. Or it could be the intermediate between DNA and proteins or RNA, like this long non-coding RNA I talked about on the X chromosome. But as I said on my last slide, the medicines and interventions, they will come. The hope is there now that we have the right targets. It's going to take time and money, but they will come in the future. And I think it's very important as a community that all of our efforts need to be focused and really on keeping these drugs when they come affordable to the families so they can access them. And lastly, as clinical genome sequencing increases the number of ASD genes identified, many of these are being found in other disorders or conditions or diseases, including ADHD, which we talked about yesterday. I talked about epilepsy, intellectual disability. And it really begs us to address this question or readdress the question of what is autism or what is autism spectrum disorder. And the definition of ASD, it's evolving all the time, and it needs to be considered in the context of the family and also the systems that they live in. So I think this is one of the reasons it's so important to share our experiences from north of the equator with those of you in Australia and have this type of a public symposium to get your feedback and input. So thank you very much.
I think, Stephen, when we look at your genome, we need to find the gene that makes you resistant to jet lag, because I've been absolutely amazed at your performance over the last couple of days. So thank you very much for the energy that you've been putting into this. Thank you. So it's often stated in the autism world that if you've met one person with autism, you've met one person with autism, and Stephen mentioned the diversity. And that means that if two individuals are living with autism, they may be as different as they are the same. And when we think about genetics, we know there needs to be robust conversations about the benefits and risks that genetics brings into this complex world that we already live in. And I think that those conversations need to be amplified in autism, where we have such enormous diversity. So it's with those things in mind that we've brought together a panel for some conversation this evening, but we've only got time for a very beginning of the conversation, and we're hoping to get you to help us to think about other ways to extend the conversation. And indeed, we've set up an opportunity for you and for you to share with friends and colleagues and family whether or not you'd like to join in further discussion about genetics and autism, and if so, what you'd like to bring. And that's accessible through the Turner website or Facebook page, and we'll go live at the end of this forum. So I would like to introduce the panel. I won't reintroduce Stephen. Professor David Amor is a clinical geneticist and researcher, and is currently the Lorenzo and Pamela Galli Chair of Developmental Medicine at the University of Melbourne, and he works as a clinical geneticist at the Royal Children's Hospital. Eliza Hill, next to David, is the parent of Jay, who has autism. Eliza also works as a laboratory researcher and is head of the Gut-Brain Axis Laboratory at RMIT. And next to Eliza is Professor Lynn Gillam, bioethicist at the University of Melbourne. I've had the privilege of working with Lynn over the last few years when I was working at the Royal Children's Hospital in neurodevelopment and disability. And I think on a daily basis, because it's a little bit hard to understand what bioethicists do for some people, both in clinical settings and in research, Lynn strives to find a balance between what can be done and ethically what should and shouldn't be done. So we're very grateful for this breadth of experience that has come to be here this evening with you. And I'm going to start because I think there may be some people in the audience who are quite keen to understand what's available for the diagnosis and genetic investigation of autism, by asking David to explain what's possible for genetic testing in Victoria today and how you might access that, just in case that's of importance to anyone here. Thanks, David.
Thanks, Katrina. And I think Stephen's pretty much summarised both the situation in Canada and also what it's like in Australia, and particularly in Melbourne. So we see quite a lot of children with autism in our genetic service. We don't see every child with autism, and I would, I guess, preface my comments by saying that our genetic testing response is very much driven by parents. We're seeing children, and we are trying to respond to questions that parents have. So the extent to which we undertake testing is very much driven by the questions that the parents have. And one of the important things there is that different families come with different questions. So with some families, they are particularly interested in a sort of broader philosophical question about why has this condition occurred in my child. Other parents are really driven on wanting to get treatments or to make sure that we're not missing a treatable condition. And for others, the question is very much about recurrence risk. If they have another child, will they have another child with autism? So the approach we take will be influenced by the questions that we're being asked. But put simply, we test nearly all of the children that we see using chromosome microarray, which Stephen has talked about, and we test them for Fragile X. And usually we also do a test called a urine metabolic screen, which is a pretty broad-based urine test, which will pick up a range of ultra-rare metabolic conditions. So that's the starting point, and certainly all children who have autism and have accompanying intellectual disability we would recommend those tests for. Occasionally we see children with high-functioning autism or autism with average intelligence or above average intelligence. Our experience is the yield of any of those tests is pretty low in that group of patients, and so we don't tend to recommend testing in that group, but once again, if the family have particular questions that we think can be answered using genetic testing, we will apply them. In terms of the next step, and that I'm particularly thinking about genomics, exome testing, and whole genome sequencing, we do have access to that testing for some patients, but we are really focusing on the more complex group, which Stephen described. So patients that have additional features apart from autism. So what I'm talking about there is the presence of significant motor delays. It looks as if just something as simple as the age of walking is quite an important determinant of whether we might find a specific genetic cause. So, you know, children that don't walk until after age 2 have a much higher chance of having a diagnosable genetic cause than those that walk at an average time. Things like facial differences, the presence of other birth defects such as cardiac abnormalities would all point to a specific genetic cause, and at the moment our exome testing really is targeted at that group. And that's partly based on funding. That's what we have funding support to do. But it's also based on the fact that we know from experience that those are the patients which have the highest chance of us finding an answer using genomics.
Thanks, David. So I've got really one question that I'm going to ask each of the panel members in turn. I'm going to start with Stephen and give him an opportunity to add to what he's already been saying. But I think we know what you're going to say. The question's very broad. Would it be a good thing to know the genetics of autism? And in particular, anything you'd like to stress or add to what you've said already in the talk?
Oh, I hope I argued the answer is yes. But in fact, we asked the families involved in our study these exact questions, and we do surveys and get feedback on those hundreds of families we delivered the information to. And as far as I know, and we've published some of this, but in every case, the families are very happy and pleased that they got information back. And it helped in some cases to help answer the questions that they wanted answered. So, and ultimately, contributing this data to the larger body of knowledge and helping other families get answers is really important to them. So the short answer is yes. I think the only thing I would add is communicating the information in an informed way back to the families and the health care providers is incredibly important. And this is the difference between places like Australia and Canada and the UK compared to, say, the US, where often in the US you don't get the data back from a genetic counselor, and there's a lot of confusion. It's very important that this goes back. It's complex information. I had my genome sequence done, and I needed genetic counseling to understand some of it. So the families need it. And the other point I would make is an interesting question is, the reason we invested so much to do the genome sequencing is the data is really good and it's static. It stays there. But when you reanalyze it, 12 months or 24 months later, you're going to find new things just because there's more data to compare against. So families shouldn't give up their pursuit of an answer because often it'll come when you reanalyze the data. And I think this is one of the challenges for the clinical labs is to have the infrastructure to do this. But it's exciting times because now we can provide some answers to the families.
Great. Thanks. Next, Elisa, who has joined us. And I'm just going to show some slides that Lisa sent through of her with her lad Jay, so you have a sense of her experience of living with Jay. It's very good of Elisa to share these with us. So, Elisa, thinking about your life with Jay, would it be a good thing to know the genetics of autism?
Yes,
Please
yes.
tell us a little about your life.
Yeah, so I mean, I think I need to put it into perspective, and I'm sure there are families here in the audience as well that may have a similar experience. So I think what Stephen talked about as that complex group, we're right in there with that. If we think of flavours of autism, we've got red hot chili. So you might not be picking it up from those photos because I'm showing you the really, beautiful ones. Just to let you know, Jay was born overseas. We came back when he was about 2. He was diagnosed shortly after that. He's nearly 16. He's actually grown since most of those photos, so he's about up here. He's minimally verbal. He's as agile as an antelope, and he's got a great laugh, and he loves music. He's got all of those other things that you saw on the slides, except seizures. He doesn't have seizures. So when I say complex, we've got those complex behaviours, and that's really changed our lives. It's really changed the way we live. You can see that I have a younger son. He's in that top, next to the right photo. He's now in secondary school at boarding school, so that we can, because of these changes. So for us, it would be really great to understand the cause of this, and perhaps, know whether there's something we can do to improve that quality of life. We've had those original tests, and I actually think they were done potentially when David was involved in those tests. We had those tests done, the basic microarray and Fragile X, and these are negative. So those things have been done. He's had early interventions, as many hours as we could do for many, many years. So all of those things have been done, but we'd still really like to know, and we think that there could be really strong benefits for us.
Thanks very much, Elisa, and for sharing your family's story. Much appreciated. David, I'll ask that same question of you. Good thing to know the genetics of autism?
Yeah, so I'm going to answer yes to that, and I guess I'll just preface that by saying that, my focus and my clinical work is very much around the complex cases and the kids with more severe disabilities, but there's no question that in the last, just 5 years or so, we have made such dramatic advances in terms of being able to give answers to families. It's impossible to overestimate the steps that we've made. All of these families where we've followed them up for years, and they've kept coming back, and we've said, oh no, we still don't really understand what's going on, and all of a sudden, family after family we're able to give specific diagnoses for, and the value that the families place on that, whether it be because it opens the door to treatment, and I'd be the first to admit that's only a minority of cases, but it is some cases, the ability to connect up to support groups, to other families on the other side of the world with children with the same disorder, advice about reproduction. We have families where they've decided not to have another child because they're so scared, and suddenly we can say, actually the first child that was affected, we've shown it was a de novo mutation, and therefore, there is minimal risk of you having another child that's affected, and they can then go on and say, well, they can then go on and have another child. So, so many of these benefits we've seen, and I guess I look forward to the time when we can offer that sort of service to even more people.
Great, thanks. Now, Lynn, your turn. A good thing to know the genetics of autism?
So a yes but, or a yes maybe. So clearly there's lots of good things. Knowing the genetics of autism is good if it makes people's lives go better, and you've talked about ways in which it could make people's lives go better, but I think there's a couple of really big challenges both for individuals and for society to think about. One of them is around the use of that genetic information in prenatal diagnosis, and Stephen, you alluded to it a couple of times talking about recurrence risk. David, you talked about it as well, but you talked about the kind of happy part of the story, which is we know that the recurrence risk is not high, but what about when you do know that the recurrence risk is high? So then people are in a position where they have to make a choice about what will they do with that information. Will you have prenatal testing? What would you do if you found that that pregnancy or that fetus also carried that genetic mutation? Because with no treatment, certainly not any treatment in utero, so does that mean we're suggesting that termination of pregnancy is an appropriate response to finding that gene in a fetus? Now that seems like a really big question. It's a big question, I imagine, for the family, but it's also a big question for society. Do we think that's an appropriate thing to do and a thing to fund. We're seeing at the moment in society quite a lot of push for lots of prenatal screening and even pre-pregnancy carrier screening. So if we could do pre-pregnancy carrier screening for the whole population to identify mutations that might cause autism, would that actually be a good idea? Because it might change who's born. Do we want that change? To what extent do we want that change? And I think the other challenge is related to that is something about identity. This question about who do we want to be born and what sort of people do we want to have comes up in relation to genes for lots of medical conditions and disabilities. But I'm wondering if in autism there's a particularly tangled connection between the, do you say the disability? And the person, the personality.
This is fraught with difficulty, actually, this terminology, and depending on who in the community you're speaking with. And so certainly at the higher functioning end, it's not considered a disability at all. There are just glowing examples of such great ability. But I can speak personally that we certainly are looking at disability with intellectual disability and minimal verbal interactions. So to put it into perspective, it's not just autism and disability.
So one of the questions I guess I'm wondering about is if you could change the intellectual disability, if you could change the verbal communication, is your son still the same person? That's a question that people have raised. Would we actually be changing the identity of the person? And I think it's a really big question about what makes someone who they are and what makes them special, how much you can change them before they're not the same person anymore. So whilst I'm not suggesting at all that all of this is bad, I do think it raises some big questions that we need to think about, both as individuals but as a community.
Thank you. It's after 7:30 and I think that was our time to finish. So I'm going to wrap up without an opportunity for questions, and I apologise for that. But please do visit the Facebook page and offer suggestions about points that you would like discussed, indicate your interest in joining in discussions, provide your email addresses or other contacts so that we can get in touch with you. And we're really hoping that, having had the opportunity to hear from the North, thanks again, Stephen, fantastic to have you here, and to hear some facts and different perspectives on the issue of autism and genetics, that we'll be able to generate further conversations that are going to really push us to be doing the very best for individuals in this complex society in which we live. So thank you very much for coming. Very much appreciated.